Okay, well, welcome to tonight's webinar. I'm Dr. Jenny Tullett, and I'm going to be talking to you about some of the things that I teach about and research about at the School of Biosciences at the University of Kent. We actually work with this third group of worms here called Caenorhabditis elegans. Okay, and these are tiny little things. This is the lab mascot, which is a bit bigger and stuffed, but these are the worms that we work with. And actually, even this big one here in the middle, it's big because it's taken under the picture is taken under a microscope, but that is actually only 1 millimeter long in real life. Okay, and this is an adult worm. And inside this adult worm, you can actually see just about some embryos. Okay, and each adult worm during its life lays around about 350 of these embryos onto the plate, and you can see those, they're these oval shapes here. And these embryos hatch and they develop into larvae, and the larvae go through 4 stages, L1, L2, L3 and L4, before they come to be an adult themselves. And they're hermaphrodites, so they're self-fertilizing organisms. Okay, they both produce eggs and sperm. And they're made up of 959 cells. And the position and the function of all of these cells is known, which is quite amazing for something that is quite complex, even though it's very small. There's 20,000 protein coding genes in the worm. So that means there's basically 20,000 genes that make something that is useful to the worm. Okay. And of those, about 75% of the worm's genome is actually conserved to humans as well. Okay, so things that we do in the worm and the way that we use this worm to understand scientific questions is actually highly relevant to human biology as well. And this is a nice, very schematic textbook image of a hermaphrodite worm. And here you can see the eggs drawn in the middle just before they hatch. Okay, and this is the head of the worm here, and this is its tail. At the moment, one of the things we're doing in my lab is to think about the question about why we eat or when we eat, and how do we know when to stop eating at the moment. Okay, so we're using these worms to ask the question, how do animals perceive or sense or react to food? And this is really important in biology because these kind of responses have implications for a whole range of different metabolic disorders. So things like obesity or eating disorders or diabetes, all of these things can be impacted on how we react to food. So as you were quite rightly saying in the poll, there's actually a whole range of different things that are required for a body to actually be able to sense food. Okay, so you obviously smell food, you taste it, you see what it looks like, and that tempts you to eat it. But it's far more complicated than that, really, because you actually also have signals from your digestive system, which are known as gut peptides. So things like PYY and GLP-1, all sorts of things like that. Ghrelin you might have heard of as well, and they come up from the gut. You've also got signals from other tissues. So for example, your fat tissue, you might recognize the hormones insulin or leptin, you might have seen in stories in the news or learned about in school. But all of these signals are actually integrated in a part of the brain called the hypothalamus. Okay, and this is required for regulating a process called satiety, which is basically how you know when to stop eating. Okay, and all of this stuff comes together and is processed by the brain. And we call this neuroendocrine control. Okay, and this sounds like a bit of a fancy word, but you can basically break it down as you can with most biological terminology. So neuro basically means brain and endocrine means hormone. Okay, so you've basically got brain hormone control of learning how to eat and when to stop eating. And it's actually possible to study this in a worm. And one of the things we do is we study the genetics of it. So the genes that we have in our bodies that are responsible for controlling food-related behavior. And we can do it in something simple like the worm, because the worm does actually have a few different simple, measurable, scientists like things that can be measurable, food-related behaviors. It's got a genetic system. We know all the genes in its genome. the 20,000 protein coding genes I was telling you about, and also its whole nervous system. So all the neurons in its body have been mapped. We know where they are and we know what most of them do as well. Okay. And the work I'm going to tell you about today was the work of Nick, who's a PhD student in my lab, and Max and Alex, who are also students in my lab a little while ago. And actually, it's interesting to see that both Nick and Alex were actually undergraduates at the University of Kent before they started their work with me. Okay, so if we're going to use the worm to study food-related behaviour, it's really, really simple. You've basically got food intake. So at the mouth of the worm, the food goes in. There's a whole range of chemosensory neurons. These are kind of neurons that sense food or sense the food in the environment before it comes into the worm. There's endocrine signaling. So things like insulin hormone, they signal to the rest of the body of the worm. And all of this comes together again in the nervous system to control the behavioral change of the worm. And those behavioral changes in turn feed back to control the food intake. And worm genetics is really very simple. We learn about it in our degrees, but there's really only two concepts you need to grasp to understand it. The worm is a really good genetic model. Okay, and this is a way we use terminology of just saying that something's really good with doing genetics. You can do really good genetics with it. Okay, so you can do two things. You can either knock genes out, so you can take genes away, or you can add genes in. Okay, and by doing that, you actually learn an awful lot about what different genes do. Okay, and then you can get a little bit more fancy if you want to, and you can actually knock different genes out at the same time as each other, and you can actually find out how genes work together as well. Okay, but for the sake of today, you need to know is that you can knock genes out and you can stick them back in, and that tells you what the gene does. Okay. So, next bit of knowledge that you need to know is that worms have different food-related behaviors. Okay, they basically do three things, right? They roam around a plate, they look for, and while they're doing that, they're looking for and eating some of the food. Or they kind of start to sit still and they do this thing called dwelling, where they move back and forward a little bit. Okay, and that's when they're eating the most of the food. Or they do something called quiescence, which is basically sleeping. And this is the thing they do when they're completely full. Okay, so it's related to human satiety. Okay, and we can measure these things in a worm really easily. Okay, we've got a lovely assay that we do. You stick one worm on a plate of food, okay, before you go home in the evening. Okay, and then you come back 16 hours later, right? And you take a photograph of the plate, and you can see all the places that the worm has visited on that plate overnight. Okay, so this is the nightlife of the worm. And then you can put that photograph onto the computer and you can put a grid over the back and you can work out what percentage of the plate the worm has walked over. Okay, it's really neat. And this is the kind of picture that you get when you take these images. Okay, so this is a normal worm. Okay, this is what a normal worm does. And a normal worm covers around 70 to 80% of the plate overnight. Okay, so we've been interested for a very long time in our lab in a gene called SKIN1, SKN1. Okay, and this gene is a transcription factor. Okay, so what that means is that SKIN1, this gene, once it's been made into a protein, it can stick to the DNA. Okay, and what it does is it switches on other genes. Okay, and together, all these genes that switches on do all sorts of interesting things in the body. Okay, they're involved in the stress response. They can balance the redox state, so the amount of oxygen that's available. They control fat, mitochondria function, immune responses, and a whole range of other different things. Okay, and as a result of that, SKIN1, this gene that we're interested in, is really important for regulating lifespan. It regulates stress resistance. And for the purpose of today, it's really important for food sensing behavior. Okay. And importantly, this gene is also controlled a little bit by the environment, and specifically the diet that the animal has. And it's interesting, not just because it's a gene that we're studying in the worm, but because mammals also have a version of this gene, and it's called NRF. And obviously in an ideal world, the gene name would be exactly the same in the worm and a mammal, but unfortunately that very rarely happens in science, okay? So you just have to remember SCIM1 is the same as NRF in mammals, okay? So we're interested in this gene and its role in food-related behavior. Okay, so there's 4 things called isoforms of this gene. So that basically means there's 4 bits of DNA in the worm that code for this gene, and they're all slightly different sizes. That's all you need to know. And we're interested in one of these, this little one here, okay? And remember our genetic model, okay? So you can knock genes out and you can put them in, okay? So if you knock genes out, we basically make a mutant, okay? And we have a SCIM1B mutant, and that mutant basically means that you've taken out a chunk of DNA, okay, in this isoform here, and that means that this gene can't be made anymore, okay? So that's a mutant version, and that allows you to understand what the gene is doing. And you can also add genes back in, and if you add them back in, you can do something pretty cool. You can basically just tag them with green fluorescent protein from those jellyfish I mentioned earlier, okay? And if you tag this little bit of gene here, this SCIM1B gene with green fluorescent protein, you can see that you get these 2 little spots in the head of the worm, okay? And these are really cool. And they're called the ASI neurons. It basically looks like 2 eyes, actually, when you're looking down the microscope. But because we know the structure of the whole nervous system of the worm, we know that these 2 neurons are actually called ASI neurons, okay? And these neurons are known to be chemosensory. So that means they are actually capable of detecting food in the environment. So that was a really cool experiment to do this, because we actually know where this gene is in the worm. And often when you know what bit of the worm something's expressed in, it tells you a little bit about what that gene might do. So for us, that was giving us a clue that it might have something to do with food type of behaviors. Okay, so the question we asked was whether this gene was actually controlling behavior in the worm. So it's only in these 2 neurons, right? And if you've got a normal worm, this is what happens if you put a worm on a plate overnight, okay? It's quite busy. You can see it's been roaming around quite a lot. However, if you have a type of worm that doesn't have that SCIM1B gene in anymore, look what happens to its behavior. Look, it can only cover about 40% of the plate, and all its little tracks are really convoluted and tied up together, okay? So why do you think this is? Okay, let's try the poll again, see if I can do it right this time. Okay, so 3rd poll. Okay, so why do our mutants move less? Is it because there's something wrong with their muscles? Or is it because they show different behavioral patterns? Or do you think it might be both of those things?